Okay, so today we're going to be talking about the first part of blood vessels, and the other two parts of blood vessels have already been posted on your Blackboard, so make sure that you look at those. So there are three main types of blood vessels. I've already mentioned them when we talked about heart. These three types of blood vessels include arteries, which carry blood away from the heart. Then we have veins, which carry blood towards the heart, and then capillaries, which are sites of exchange of materials. And when I say exchange of materials, I mean that oxygen and nutrients are going from blood to the tissues, and then wastes and carbon dioxide are moving from tissues into the blood. While the capillaries are the smallest vessels and only consist of single layer of thin endothelium, arteries and veins are going to contain layers that we know as tunics. So we don't really, with capillaries, we don't really talk about tunics, but with arteries and veins, with these larger vessels, they have layers which we call tunics. These tunics are tunica intima or interna. You might see either of these terms depending on where you're looking, and that's the internal part. Then the medium part is tunica media, and then the outside layer is your tunica externa. The internal space, and let me see if I have a picture of that. Nope. The internal space that's where the blood is located is known as lumen. So the inside of the blood vessel is lumen, the space. So I want you to look at this picture right here. So you can click on that picture on your Nearpod. Now, if you're not on the Nearpod, but you're just watching this on the screen, then you can't click on the picture. But I'm gonna go ahead and click on it so you can look at it. And if you're in the Nearpod, I would like you to look at these two and try to list some of the differences. What are some differences between artery and vein? Artery is the one on the left and vein is the one on the right, the one with the blue outside part. I just wanted to summarize the differences. I'm going to go ahead and pull the picture up again here. So look at it as I'm talking about this. So arteries do have thicker walls, and they also have a smaller lumen for that reason than veins. So for this reason, veins actually tend to collapse when they're not filled with blood because their space is larger and the outside is thinner. If there's not enough blood in the veins, they do collapse. The veins have valves, and these valves are going to prevent backflow of blood. These valves are the same as your aortic semilunar valve and pulmonary semilunar valve. So the valves that we were talking about in the heart, this is what they look like, okay? And here you can just see two of those cusps, but there's a third one, okay? You don't see the third one because it's the vein is cut open here. But there would be a third like little pouch, and in the middle, they're not connected, right? So as the blood is going, imagine the blood flows through the vein from the bottom towards the top. It is able to squeeze those pouches against the walls of the vein, and blood passes through. Well, when you release that, when the pressure is released and the blood would want to fall backwards, those pouches, the valve will capture or trap this blood, and as the blood fills these pouches, really, they're going to fill up, swell, and rest against each other, so the blood doesn't fall back the wrong direction. Okay, so that's kind of think about it whenever you're looking at the aortic and pulmonary semilunar valves as well. This is how they work. There's not much effect from the heart anymore in the veins, and that's why so your veins are actually going to rely on the surrounding skeletal muscle that is around the veins. So when your muscle squeezes, it squeezes the vein, and that's what pushes blood through the veins. So we have these valves to make sure that the blood is only pushed in one direction and not backwards. Veins don't have elastic lamina. So their elastic fibers are dispersed throughout the tunics. And if you look at table 20.1, you can see some like less obvious differences that are listed in that table as well. So in your textbook, table 20.1 lists all these differences. Some are not super obvious from this image, okay? But veins do not have the elastic lamina. When we look at the labeled blood vessels, so here we're looking at labeled artery and vein, you can see that the tunica intima is composed of simple squamous epithelium there on the lumen side. So the lumen is lined by simple squamous epithelium. And then there is a basement membrane. So as we're going from inside out, we have simple squamous epithelium. And this is known as endothelium. And then a basement membrane is the next layer. The basement membrane, they're labeling here as the subendothelial layer. That's the same thing as basement membrane. So those two terms are the same. And then the third layer of your tunica intima would be the internal elastic lamina. And I'm not sure, like if I was the one that made this picture, I would make the bracket probably where the layers are on top of each other, just so you can see it a little better. That tunica intima artery is made of endothelium, basement membrane, and the internal elastic lamina. So it's really hard to see on that picture that there's like that little tiny bracket, and it's hard to see that it also includes that internal elastic lamina. On the vein, you can see that we don't have the elastic layer, but we still have the endothelium and the basement membrane. So on the vein, tunica intima has endothelium and basement membrane, but no elastic lamina because there's not a distinct layer in veins usually. In most veins, there is not. So that's our tunica intima. Tunica media, if you look at the artery, tunica media has a pretty thick layer of smooth muscle. That's the red part right there. So you can hopefully recognize the smooth muscle cells. And then on the outside, as we're working our way towards the outside, you have the external elastic lamina. And so that's tunica media. On the vein, because we don't have the elastic lamina, on the vein, the tunica media is only the smooth muscle. So it's the same for the type of tissue as and the tunic. So if you're supposed to label the vein, then pay attention to what answer choices you have, or what is it asking you? Like, is it asking you to identify the tunic, or is it asking you to identify the type of cells? Okay, so tunica media in the vein is the smooth muscle layer as well. And then tunica externa, the third layer, the outermost layer, this is going to be thicker in the vein actually. And tunica externa does not have any sublayers, so it's just a tunica externa. Tunica externa contains what we call vasa vasorum. So as you see underneath that, the term tunica externa, you see vasa vasorum. Vasa vasorum is not a layer. Vasa vasorum is the little blood vessels that are found in the tunica externa. So you can see the little blue and red dots in that tunica externa. Those are little tiny blood vessels. And this is because the blood that flows through the arteries and veins is just simply passing through them. It does not bring any nutrients or oxygen to that vessel. The vessel is just a pipe. But vessel is made out of cells, and cells need oxygen and nutrients, and they need their waste products to be taken away. And so that's what the vasa vasorum does. It supplies the cells that make up that blood vessel. So if you memorize the layers on the artery, then the layers on the vein are going to be the same, but you just take away the elastic layer. Okay, the elastic layers are not there. And I like to remember it for the artery as 3-2-1. So the innermost layer has 3 sublayers, the medium layer has 2, and the outermost layer is just 1. Okay, so as far as sublayers go. And then, if you just remember that, then remember that your vein does not have any elastic lamina, but the order is going to be the same. What we are actually looking at here right now is we're looking at maybe like a medium vein. And then the artery here, this is a muscular artery, okay? And I'm saying that it's a muscular artery because we are actually going to have different types of arteries. So I was gonna have you try to label this if you remember. So on this one, you're supposed to label the parts of the artery. It might be easier if you're doing this activity. So try it and see if you can remember. You can have your book next to you and just kind of do it to reinforce the information. I think it's the easiest if you have this pulled up on a tablet, use a stylus and write it in. It's kind of difficult to write with your finger or with the mouse if you're on a computer. If you're on a computer, you might want to add like a text box in there and just type it in. And that's an easy way to do this activity. Okay, so we're looking at this. We can go over this together. So going from top to bottom, the first three closer to the artery are going to be endothelium, then basement membrane, and then internal elastic lamina. Those three together are going to make your tunica intima. And then the two below that are going to be the smooth muscle and external elastic lamina, and those two together make the tunica media. And then the last one is tunica externa. Okay, so that's how it should be. And I'm gonna move over here. I have another one, so you can do that one on your own, but this would be for the vein. So same kind of the same thing, but for the vein. So try it, practice it yourself whenever you're studying. So let's talk about the types of vessels, okay? So, the types of arteries and types of veins. So there are different types of arteries and veins. We just mentioned like the different types of vessels, but now we're looking at the group of arteries and the group of veins, and we can distinguish them based on size, and they're also going to differ in their layers. So these different types of arteries actually have specific names. With veins, veins are kind of boring. So it's just like large veins, small to medium size, and venules are the smallest ones. But arteries actually do have names. The largest arteries are going to be elastic arteries. These elastic arteries include aorta, they include pulmonary trunk and their branches. So as far as numbers go, we don't have a lot of these largest vessels. There are the biggest ones, but we only have a few of them. These arteries have elastic fibers dispersed all through the tunics. So they do not have an elastic lamina like we saw on the muscular artery. And that's the one that we were labeling just a little bit ago. So elastic arteries are called elastic arteries, but they do not have elastic lamina. Write it down somewhere and highlight that. So elastic arteries are the largest arteries. They have elastic fibers in all layers, but they're not concentrated in the distinct layers. They're just everywhere. So the whole thing is like very, very elastic. And when you look at that elastic artery, when you look at that picture, you can kind of see like these yellow fibers, and that's what's supposed to represent those elastic fibers. These arteries are known as conducting arteries, and the type of the artery that we labeled in the sublayer, where we labeled all the sublayers, that's your muscular arteries. So these elastic arteries eventually will branch into muscular arteries. These are smaller. These are still pretty stretchy, but they're a little less stretchy than the elastic arteries. They have thicker muscularis, that's why it's a muscular artery. And it allows, this muscularis allows these muscular arteries to vasoconstrict and vasodilate. So they are able to change the size of their lumen based on their based on the needs. The elastic fibers are concentrated here within the internal and external elastic lamina, as we saw on the image when we labeled the artery. And we call these also distributing arteries. So these would be going towards a specific organ, okay? These arteries, these muscular arteries are going to branch into smaller arterioles, and arterioles can vary quite a bit in size. So there is the biggest variation in the arterioles that we see. You can have some larger arterioles, and these larger arterioles are going to have all three tunics, intima, media, externa. But by the time the vessels branch into the smallest arterioles, right before the capillaries, you will only see the endothelium, basement membrane, and a single layer of smooth muscle. And we will see that when we look at the capillary exchange here in a moment. Okay. So largest arterioles have all three tunics, but by the time we get to the smallest one, they only have the endothelium, basement membrane, and single layer of smooth muscle. The blood from these arterioles will then flow through capillary bed. And then as the blood flows through the capillary bed, eventually it will be collected by venules. And venules are your smallest veins. The venules merge into, into larger, into larger vessels, which are going to be your small to medium veins. And then those medium veins will eventually merge into large veins. Largest veins are our inferior and superior vena cavae. Okay, so those are our largest ones. So that's the types of vessels. Yeah, so that's that. Okay, let's talk about types of capillaries. So we had types of veins and arteries. We also have different types of capillaries. The three types of capillaries you can see on this image right here. There are about 10 billion capillaries in our body. And if you were to connect all of these together, it would add up to about 60,000 miles. That's kind of an estimate. I have seen numbers that vary by, 10,000 miles up or down before, but most literature kind of agrees on that 60,000 mile length of capillaries in our body. This is also 100,000 kilometers if you want a metric system there. There are different kinds of capillaries. So continuous capillaries are completely lined with endothelium. They're surrounded by a basement membrane. That's a complete basement membrane. Most capillaries in your body are going to be continuous capillaries. So most of them are these ones, the continuous ones. The next group or next type are your fenestrated capillaries, and fenestrated capillaries are just like continuous, except they're going to contain fenestrations or pores in the endothelial cells. So you can see that the fenestrated capillary looks like it's kind of dotted, okay? Those dots are fenestrations. They're these almost openings in the cell membrane. And these openings are going to allow larger things to exit that capillary, okay? Continuous capillaries are pretty restrictive as far as what can exit the blood vessel. So they keep most of like larger things in the blood, and only fluid and soluble things and small things can pass through. But fenestrated capillaries, due to these fenestrations, allow maybe some small proteins to exit or enter the blood. And we see fenestrated capillaries in kidneys, also in the small intestine, and most endocrine glands as well. So you see them in places where you also want to like maximize the transport in and out of the blood, which, kidneys would be it. Small intestine, this is where you're absorbing all the nutrients. So so having the fenestrated capillary is more beneficial for increased absorption. And then endocrine glands for passing the hormones. The last type of capillaries are sinusoids, and sinusoids have large gaps between cells, and they also have incomplete or absent basement membrane, as you can see on this image right here, that right side. They are found in areas where formed elements and large particles need to enter or exit the blood. So for example, red bone marrow, where you formed elements, the liver and spleen, where we recycle formed elements. In order for the newly created erythrocytes or leukocytes to get into the blood, we have to get them somehow into the blood, right? And so these sinusoids, having these large gaps, allow for erythrocytes and leukocytes to enter the bloodstream. But you don't want, like in your regular tissues, you don't want the erythrocytes to be leaving your blood. They need to stay in the bloodstream, right? And so you would be seeing them in red bone marrow, liver, and spleen, okay? These sinusoids here. Actually, when we'll be looking at spleen in third unit, okay? So when we talk about lymphatic system, and I will mention the word sinusoid, and when you will hear it and when you will see it, you'll be like, wait a minute, that doesn't even look like a vessel. So sinusoid is almost like a hybrid between like a blood vessel and a space kind of thing. So because of those large gaps. Okay. So that's our capillaries. Now let's talk about some capillary beds. Capillary beds are collections of several capillaries, anywhere around 10 to 100. So some smaller capillary beds have only about 10 capillaries, kind of interconnected, but large capillary beds can have up to like 100 capillaries that are all interconnected. These capillaries are going to function together in capillary exchange. Each capillary bed is supplied by a precapillary arteriole. So you can see that on the actually, you can't see the title there, but it's the red vessel that goes up and down. And that's your precapillary arteriole. And from it branches a mid-arteriole, okay? So that kind of a middle vessel there, it looks like you have a red vessel and a blue vessel, and then in the middle, you kind of have like the main mid-arteriole on the arterial end, and then you have the thoroughfare channel at the venous end, okay? So this vessel that's kind of connecting the two, the arterial and the venule, at the arterial end, it's called mid-arteriole. At the venous end, it's called thoroughfare channel, okay? And out of this, we see branching of true capillaries. Okay, the true capillaries branch from the mid-arteriole, and they kind of loop, and then they will connect to the thoroughfare channel at the venous end there. At the origin of the true capillaries, you see little pink, it looks like little pink loops. These are smooth muscle cells, and we call these precapillary sphincters. These precapillary sphincters can regulate blood flow into the capillary bed. When they close, blood goes straight through to the thoroughfare channel, and when they're open, blood flows through the vessels. So at the top picture, you see the opened precapillary sphincters, so blood is allowed to flow through the capillary bed. At the bottom picture, you see what would happen if these precapillary sphincters would close. And if they close, then the blood just passes through and goes into the venule. This is, since there are about 60,000 miles of capillaries in our body, we only actually have about 250 to 200 milliliters of our blood volume in the capillaries at any given time. So, and our total blood volume is about 5 liters, right? And so due to the amount of capillaries that you have in your body and the amount of blood that you have, there simply isn't enough blood in our body to fill all the capillaries in our body with blood at the same time. And so capillary beds take turns. Some capillary bed is going to be closed while another one next to it is open, and so on. Actually, only about 5% of capillaries in your body has blood flowing through them at any given time. But again, they alternate, so they don't stay closed for very long. We will also be talking about like redistribution of blood flow. So you'll see that tissues that are more metabolically active are going to have more capillaries open than tissues that are less metabolically active. So precapillary sphincters contract rhythmically about 5 to 10 times each minute, okay? So that's how the capillary beds work. This is just showing you the distribution of blood in our body. So you have about 18% of our blood volume is going to be in the pulmonary circulation, About 12% of our total blood volume is in the heart. And then 70%, most of the blood in our body is in our systemic circulation. If we take this even further and look at the systemic circulation distribution, we'll see that arteries only have about 10% of blood of that 70%. And so it's 10% of the total blood volume. Systemic capillaries, about 5% of our total blood volume. And the most of our blood is located in the systemic veins. Okay, so that's an important thing to know as well. Know where most of the blood is located, and it would be in our systemic veins. And this is because our veins, remember, veins have the larger lumen, so they're able to accommodate more blood than the arteries with their smaller lumen. You will be looking at in the histology file, which, just as a reminder, histology file is in the Science Learning Center in the Anatomy and Physiology resources. And that's where the slides are. You have an artery and vein slide. So you will see artery is going to look nice and round and very thick with a small inside. Vein is going to be like super squished. It's not even going to really resemble like an oval or anything. Your textbook is actually giving you a very nice picture of vein. It looks almost circular, but the picture in my slides in that histology file is going to have the vein much squished. So be aware where the blood is located. I'm not really going to ask you about the exact numbers, but be aware where is the most and where is the least blood volume in our body. Okay. We'll talk about pathways of blood, and then we will talk about the cross-sectional area, and then we'll get into capillary exchange. Okay, so the pathways of blood in our body, we can describe them as simple or alternative pathways. Simple pathway is where a single major artery supplies a tissue or an organ, and a single major vein leads blood out of that organ. Example of a simple pathway would be spleen. You just have one artery, one vein supplying that organ. Alternative pathways are going to include anastomoses and a portal system. So anastomosis with the SIS ending, that's a singular version of this word. SES, anastomoses, would be plural of the word, just FYI. The anastomosis means connection between blood vessels. We can talk about arterial anastomosis, and this is where several arteries supply the same region or an organ, like superior and inferior epigastric arteries are an example of arterial anastomosis, or circle of Willis in the brain. So if you have looked at the vessels yet or already, this might be familiar here. But circle of Willis, when you see it, that's definitely an arterial anastomosis. There are anastomoses between coronary arteries also, but these are so small, they are not really functional. And for that reason, the coronary arteries are considered functional end arteries. And what that means is those connections are so small, if one side gets occluded, the remaining blood vessels are not able to supply the heart sufficiently. And so that's why we call them, even though they're not really end arteries, we call them functional end arteries because of that fact, that if one gets occluded, the remaining connections are not strong enough to keep the heart healthy. The venous anastomoses are more common than arterial anastomoses, so we see more of them in our body. And this is when several veins are going to drain the same region. Basilic or brachial and cephalic veins draining the upper limb are an example. Also, your dorsal and venous arch of hand and foot are an example of venous anastomosis. And then you see the arteriovenous anastomosis, which is a shunt. Shunt involves a complete bypass of a certain region. So instead of going into that area, you're going to lead the blood around it. Okay, so that's arteriovenous anastomosis. It's connection between an artery and vein directly, skipping the capillary bed. And then portal system is where you have two capillary beds and they're connected by a portal vein. We've already seen a portal system in chapter 17. Do you remember what it was? It's kind of a long term. It's like 2 words connected with a dash, portal system. Okay, think it to yourself. I'll tell you in a second. Starts with an H, portal system. So it's the hypothalamo-hypophyseal portal system, and this was associated with the anterior pituitary, right? And so you had that primary plexus that was your first capillary bed that was receiving the hormones from the hypothalamus. And then we had 2 portal veins, and those brought the blood to the secondary plexus, and that was around the anterior pituitary. So it's a very important portal system. It's also very small. We have a much, much bigger portal system in our body as well, and that's your hepatic portal system. And this is where the blood that's coming from your digestive system, it's going to drain into a hepatic portal vein. Then hepatic portal vein is going to go to liver, where it branches through the liver. The blood flows through the liver, gets processed. The liver does its magic on the blood and kind of cleans up and filters that blood, metabolizes things, and only then we send that blood to the main circulation. Okay, so that's a portal system on a much, much bigger scale compared to that hypothalamo-hypophyseal portal system. So that is portal system. So these are our different pathways here. So again, if you hear anastomosis, think about connections between vessels, and then we can have different types there. So let's go and see what we have next. Oh, I already asked you that. I forgot I had it as a question. So going on, let's talk about this total cross-sectional area of a vessel and blood flow velocity. So this graph is simply showing a relationship between the total cross-sectional area of a vessel type and blood velocity. A velocity is like speed. So think about blood, like how fast is the blood moving. When I say total cross-sectional area, I mean it's a cross-sectional area for all the vessels of that particular type. So remember in the beginning when I told you we have your elastic arteries are the largest ones, right? They have the biggest lumen, but we only have a few of them. So if you took the cross-sectional area of an aorta and then cross-sectional area of a pulmonary trunk, and then some of those other ones that are elastic arteries, you would have something that's not super big all combined because there's only a few of those vessels. But when we look at the capillaries, which are the smallest ones, their capillaries, the diameter is about as big as an erythrocyte, just big enough so erythrocyte can squeeze through. But we have 60,000 miles of them. We have so many of them in our body that if you add the tiny little cross-sectional area of every single capillary in your body, it's going to be so much bigger than the cross-sectional area of those few large vessels that you have. And so the largest vessels have the smallest total cross-sectional area, so combined. The smallest vessels, your capillaries, have the largest total cross-sectional area, okay? So that's what the blue line is showing you. The red line is showing you the speed of the blood in these vessels, okay? So in the elastic arteries, our largest arteries with the smallest combined cross-sectional area, the blood flow velocity is very high, okay? So blood moves very fast through these large elastic arteries. And why is that? Well, think about this. These are the vessels that lead out of the heart. So they're receiving blood as the heart squeezes that blood into that vessel. So that's going really, really fast because the heart is pretty strong. Okay, so when it squeezes, it just shoves this blood into this vessel and the blood moves very fast. But as the blood vessels begin to branch, there's going to be an effect of resistance and also effect of the smaller diameter. The velocity is going to decrease. So the blood is actually going to slow down a bit in the smaller vessels. And when it slows down, what also happens with it is that the pressure decreases as well. Okay. I want you to think about like a garden water hose. If you turn the water on as high as you can, the pressure is pretty high, right? In the water, the garden water hose. But if you only turn it a little bit, if you have, if the blood flow, the water flow is slower, the pressure is going to be lower too. And so in these capillaries, the velocity of the blood is the slowest. And also the pressure is the lowest because capillaries are super tiny, super thin. So you don't want them to experience the same blood pressure as the aorta is. Okay, aorta is experiencing pressure of, if we're talking about the ideal pressure between 120 to 80 is generally the pressure that can be found there. And it can withstand even higher pressure. Okay, not saying that's healthy, but it can withstand the higher pressure. But if you had blood with pressure of 120 in a capillary, then the capillary goes bye-bye. Okay, it ruptures. It's not gonna hold because it's so thin. So the velocity decreases, and that brings with it also decrease in pressure. It also allows, the slower speed allows the capillary exchange to happen, because if the blood flows too fast through a capillary, we might not be able to absorb everything out of the blood that we need to. Okay, so that's another reason why we want that blood flow to be slower in the capillaries. You can see if you follow that red line, it's going to be picking up as the veins get larger and larger. But notice that in the veins, the blood flow velocity never reaches as high as what we saw in the elastic arteries. And anybody wants to guess why the veins never reach that speed? So I want you to think about what is the main force of blood in those elastic arteries.
The heart beating.
Yes, it's the heart. It's the heart beating. Okay. So because the heart is so strong and just actively shoves that blood into the elastic arteries, the velocity is very fast. In the veins, however, we don't really feel that strong of an effect from the heart anymore. How does the blood move in the veins? It's that skeletal muscle that is going to push or squeeze those veins and just kind of squeeze the blood through. And so that force is just never as strong as the force of the heart is. And that's why you never see that velocity go up as high as in the arteries. Okay. So that's this. Now I wanted to do something else. So now we have to talk about the capillary exchange. And capillary exchange, before I start drawing it, I'm going to draw something, but and bear with me because I'm not all that great at drawing. So, but capillary exchange is the movement of nutrients and waste and respiratory gases across the capillary. And so between the blood and the capillary and the tissues, the cells that make up a tissue, right? Lipid soluble things are going to pass through the cell membrane just normally. Water soluble materials might rely on facilitated diffusion or vesicular transport, such as exocytosis or pinocytosis or endocytosis. So some substances move this way, but majority of substances are going to move by what we call bulk flow. And bulk flow refers to flow of plasma fluid. Okay, so that fluid part of the blood without the proteins from the blood through the intercellular clefts, or these are the spaces between cells. So think about, even if it's a continuous capillary, it's going to have these intercellular clefts between the cells, and that's going to allow the fluid from blood to exit the capillary, flow around the cells in the tissue, and then return back into the blood at that thoroughfare channel area. There are several pressures that are involved in this movement. So I'm gonna try to simplify. There's this picture in your textbook that I did not include on here. It's quite busy and it has a lot of these pressures and a bunch of numbers and a lot of abbreviations. So I'm gonna try to draw it a little simpler here. So yeah. So first I want you to think about the hydrostatic pressure. Hydrostatic pressure is the pressure of blood against the walls of the capillary. This is going to be our main drive that pushes the fluid out of the blood. Let me see how I can, don't think I can do it in Nearpod. Okay, I'm gonna do it in here. So now you see, no, yes, I enjoy you. Okay, so I'm gonna draw kind of a simplified version here of this. Okay, so here's our arterial and mid-arterial, and then through fair channel. And actually think about this as a true capillary. So instead of drawing it with in the loops and whatnot, it's easier to and simpler to draw a capillary like this. Okay, so the middle part, think about that as a true capillary instead of mid-arterial and through fair channel. So this is going to be your arterial. And this is our venule. Arterial is going to receive blood. So let's say it's going this way, and the pressure here is about 40 millimeters mercury. Okay. As this blood goes through the arterial, it will eventually enter a true capillary with that pressure of 40 millimeters of mercury. The presence of the blood in that capillary is going to cause the fluid to be pushing against the walls. Okay, again, think about a garden water hose. Okay, when you turn on the water and the water flows through, if you were to try to squeeze the water hose, you can't, because there's that water that puts pressure on the walls, and eventually in the water hose, it comes out only out of the end, right? But the capillary, think about it as like water hose with holes in it. Okay, maybe like that, at least at this arterial end. And so that pressure of the blood that's in the capillary, we call that hydrostatic pressure, and it pushes fluid out. Okay, so I'm gonna say this is hydrostatic pressure of blood. Okay. You also have something else in the, so it's relatively high, okay, 40 millimeters of mercury, and as the blood enters the capillary, it pushes this fluid out of the capillary. We know that in the blood we have formed elements, right? We have plasma, we have proteins, we have some solutes, right? So what I'm drawing here, these are the plasma proteins. Plasma proteins are called plasma proteins because they are designed to remain in the plasma. They don't want to go out of the blood. Most of your plasma proteins are transport proteins. They're transporting things through blood, but not, they don't really want to exit the blood. And so plasma proteins remain in the blood. They are not going to be part of the fluid that's being expelled out or filtered out at this arterial end there due to that hydrostatic pressure. So plasma proteins remain. The interstitial space, so like right here, you would have cells. Okay, that area has cells. I don't know why I did that, but I'm not gonna draw the individual cells, but this is where the cells are, right? And in the interstitial space, there is some protein, but not too much. And so now you see two areas. You see the interstitial space. So this is interstitial space. You don't have a lot of proteins there. And then we can see inside of the capillary, there is a lot of proteins there compared to the interstitial space, right? And so that creates the concentration difference. This concentration difference of proteins, this difference in the amount of proteins on these two sides, creates an osmotic pull of water from the interstitial space into the capillary. So think about it like proteins are like magnets for water. They want to pull water towards them. And so what we would see here is you would see a pool of water. I'm going to make this a little bigger. Okay, into the capillary. It's smaller than the red one, okay? So we call this colloid osmotic pressure, okay? So when you hear colloid, from now on, I want you to think about plasma proteins, okay? So colloid osmotic pressure is the plasma proteins pulling water towards them. So in this case, it's from the interstitial space back into the blood. It opposes, it goes against the hydrostatic pressure of the blood. But because the hydrostatic pressure is larger, you will see an overall movement of fluid out. So fluid goes out. This is known as filtration. I'm gonna get rid of this interstitial space, it's in my way. Okay, so because the colloid osmotic pressure exists, it wants to pull water in, but hydrostatic pressure of blood is stronger, and so overall fluid is gonna go out into the interstitial space. Now, when I say fluid, I mean it's the plasma without the proteins and without the formed elements. So it's just that water part of the blood that carries with it nutrients and oxygen and other stuff, okay, that the cells need. Imagine this fluid flow along and among the cells that are in that interstitial space and that tissue. It's going to like flow around them. As it's doing that, the nutrients are going to be absorbed by the cells. The oxygen is going to be taken by the cells, and anything else that's in there that the cells need is going to be taken by the cells. And as the fluid flows through, what's also happening is it's picking up carbon dioxide from the cells, it's picking up metabolic waste from the cells. And so imagine it moving kind of like from left to right on this picture, okay? It's going to be, there's going to be that exchange of materials. So when we say that capillaries are sites of exchange, this is what we're talking about. We're, filtering all that fluid with nutrients and what the cells need out. And then as it flows through the cells, it's delivering that stuff and it's picking up wastes. So because you have this filtration happening at the arterial end of the capillary bed, what happens to the volume of the blood in the capillary? You're losing it, right? As the blood flows from left to right on this image, you're filtering all this fluid out. Now you have less and less blood in that capillary. So what's happening, because now you have less and less blood, well, the amount of blood in the capillary determines the pressure. So now you have less blood in there. So now your hydrostatic pressure is lower here as the amount of blood decrease, or plasma decrease really. Now, nothing happened to the proteins. They're the same, okay? So proteins, and I can never make these two arrows the same, but the colloid osmotic pressure didn't change. Yeah, that's kind of pretty same. Okay, and you have this, the red one is still the HP, right? Hydrostatic pressure and colloid osmotic pressure. So at the venous end of the capillary, you have the colloid osmotic pressure is still the same as it was before, but your hydrostatic pressure decreased because you lost all this fluid to the interstitial space. What that's going to do, now the pull of the water into the capillary is stronger than push out, and that's going to promote a reabsorption of the fluid back in. So this is known as reabsorption. Okay, can't spell. Is it, maybe it has a P. I don't know. Anyway, but it's a reabsorption. I think it's a P anyway. And it is due to the pressures changing, okay? Well, the hydrostatic pressure changing. The colloidosmotic pressure, remember, it does not change. But the hydrostatic pressure, because now it's lower than the colloidosmotic pressure, fluid is allowed to return back into the venule or into the blood, into the capillary, and then it will be collected by venule, okay? About 85 to 90% of all the fluid that was filtered out will return into the capillary at the venous end, but not all of it is going to return back into blood. Part of the fluid that's in the interstitial space will be taken by this thing, and this is lymphatic capillary. Okay. Lymphatic capillary is going to take about 10 to 15% of the fluid that enters the interstitial space. The reason for this is our lymphatic system scans for any like signs of infection or something that shouldn't be there so that we can initiate immune response against it. And so the lymphatic capillaries continuously take samples of the interstitial space and then our lymph nodes monitor this fluid. So we'll be talking a bit more about that in the third unit, in the lymphatic system unit. Okay, so not all of the fluid will return back to the blood, and when the blood enters the venule, it is about 20 millimeters of mercury in pressure. So there's less pressure, and this is because venule is going to have larger lumen, but also you have a little bit less blood volume that you had when the blood entered the capillary. Okay, because some of it is taken by the lymphatic capillary. So that's kind of how the capillary exchange works. I am not going to ask you numbers. Okay, so your textbook has a much busier picture of this, right? If you have read the chapter, you've already seen it before, but there's like this, there's math in there, right? And it shows you that you have to add and subtract certain things and all that. That image also shows you the hydrostatic pressure of the interstitial fluid, and it shows you the colloidosmotic pressure of the interstitial fluid. I excluded those because they're so small, we can kind of just ignore them. Okay, they do exist, kind of, but are very small. Okay, so I just focused on the main one, that this is the main idea of how your tissues get nutrients and oxygen from the blood. It's by filtering this fluid out at the capillary, running it through the tissue, and then as it flows through the tissue, things are exchanged with the cells, and then we reabsorb that fluid back into the blood. Okay, so usually I end with a capillary exchange. I think when we finish capillary exchange, students are kind of like, but if you look at that and just go over it, try to draw it yourself, and really think about it several times, it will eventually make sense. If it doesn't make sense on the first try, it's fine. Okay, this is not the easiest topic. So okay, so that's all I'm gonna say today. Any questions about this stuff?